What are the two ways the cell can die? Okay, so if we come back to this picture where we started from, okay, where we talked of irreversible cell injury here, okay, I'll say the two main features there will be membrane breakdown, okay, and the second thing is mitochondria, damage to the mitochondrium, right? So what are the two things that can result from that? Necrosis and apoptosis, right? So moving on to apoptosis. Now, apoptosis is an ATP dependent programmed cell death. So when you hear ATP, just think of the term cell suicide, right? Cell suicide. So the cell kills itself. The cell will delete itself, right? So it's a form of programmed cell death. And there are three main pathways. There's the intrinsic, extrinsic, with the perforin granzyme pathway. So this is intrinsic 1, extrinsic 2, and the perforin granzyme pathway 3. Okay, and all these three pathways converge on a group of enzymes called Caspases. Okay, and what are they? They are proteases. When you hear the term proteases, they are enzymes that break down protein. Okay, enzymes that break down protein. So once they break down the protein, the cell will shrink in size. Okay, the cell will shrink in size. The nucleus, all the organelles of the cell will be degraded by those caspases. The nucleus will shrink in size and go under different changes. I'll show you the diagram of those changes. Okay, and then once the so apoptosis also leads to the formation of apoptotic bodies. So what do you mean by apoptotic bodies? Right? So once you have the cell undergoing apoptosis, it shrinks in size and then it will now fragment into different pieces. So these pieces are called apoptotic bodies. Okay, these are apoptotic bodies that contain different parts of the cell. Right? And those parts will now be phagocytized by white blood cells. So necrosis, so we're talking about apoptosis. So because the cell is killing itself and it's picked up by white blood cells, you typically don't tend to notice apoptosis unless you are it doesn't cause a lot of inflammation, doesn't lead to it doesn't signify that something. What I'm trying to say, it can be physiologic or pathologic apoptosis, right? It can be physiologic or pathologic or pathologic. So we'll look at those different scenarios. But when you hear of necrosis, necrosis is always pathologic. All right. So when you look at the cell that is undergoing apoptosis, what do you see? It's a deeply eosinophilic cytoplasm. So let me show you an image of that. Okay. So this is what an apoptotic cell looks like. Look at it in the middle here. So this is what it looks like here. So you can see it has retracted from all these other neighbors. These are different normal cells, right? These are normal cells, but this is the apoptotic cell here, this one. So you can see it has retracted from the neighbor, it has shrunk in size. The cytoplasm, two main parts of the cell, the nucleus and the cytoplasm, all right? So the nucleus becomes very red. That's what you mean by deeply eosinophilic. Eosinophilic means red. Okay, so it becomes deeply red, right? And then the nucleus also begins to shrink or becomes dark staining. So you can see the nucleus of this cell appears darker than the nuclei of the other cells as well. So this cell is about to undergo apoptosis, and after a while, it will disintegrate into bits and pieces called apoptotic bodies that will be picked up by the macrophages. All right. So that's what we mean by deeply eosinophilic and basophilic nucleus, which the nucleus will now also disintegrate, called pycnosis and chirorrhexis. Okay, so when you say pycnosis, that's the nucleus shrinks further in size and shape. Okay, becoming like a dark, small, blue staining ball within the cell. And then chirorrhexis means the nucleus will fragment into tiny bits and pieces, right? So moving on to the intrinsic pathway. So when you talk about intrinsic, it involves the mitochondrion, right? So I mentioned earlier there are three pathways: intrinsic, extrinsic, the perforin granzyme pathway. So now looking at the intrinsic pathway, it is involves the mitochondria, as you can see here, right? So there's a substance inside the mitochondrium called cytochrome C, right? There's a substance inside the mitochondrium. Mitochondrium is the powerhouse of the cell from our biology. It's the powerhouse of the cell. Why? Because that's where ATP is produced. That's one of its main functions, right? And how is that ATP produced? By the use of the electron transport chain, right? From your biochemistry, use of the electron transport chain. And one of those components of that electron transport chain is your cytochrome C. Now, this cytochrome C is only found in the mitochondria within the cell. No other part of the cell has it, just in this mitochondria. And inside the mitochondria, it has a very important function of participating in the electron transport chain to produce ATP. But once it leaks out of the mitochondria and into the cytoplasm, this is cytoplasm, right? Once it leaks out into the cytoplasm, it's very dangerous for the cell because it will now activate these caspases, which I said they are a form of proteases that will digest all sorts of organelles and structures within the cell and eventually lead to cell death by apoptosis. So the intrinsic pathway involves the mitochondria, and within the mitochondria involves cytochrome C. And that cytochrome C is not supposed to be outside the mitochondrium. Once it leaks out, it will activate caspases that will degrade that cell and lead it to die by apoptosis. So what are the pathways? What are the things that control that cytochrome C and keep it inside the mitochondrium? Okay, uh, that's what we talk about some BCL family of proteins, right? So those BCL family of proteins, there are two main groups. That family, there are two main arms of it. There's the pro-apoptotic. So when we say pro-apoptotic, we mean the ones that favor apoptosis or cell death by suicide, and the anti-apoptotic proteins of that BCL2 family. So when we say anti, the ones that oppose apoptosis, right? So the pro-apoptotic members are the backs and the back, all right? And then while the ones that are anti are the BCL2, BCLXL, right? So these two are in constant, they antagonize their effects depending on the state of the cell, all right? So if the cell, like when we talked of cell stress injury. So for example, we're looking at atrophy, for example, if the cell, your skeletal muscle loses its nerve supply, right? We say atrophy involves two things that happen: decrease in cell size as well as cell number. And part of the process that decreases the cell number is death by apoptosis. So if you have that scenario where the skeletal muscle doesn't have its nerve supply, right, that will stimulate the pro-apoptotic members, that's the backs and the back. And how do they affect the mitochondrion here? The backs and back will drill holes in the membrane of the mitochondria, allowing cytochrome C to leak out from the inner mitochondrial membrane out into the cytoplasm, right? Like say to bore holes in the mitochondrial membrane and allow cytochrome C to leak out into the cytoplasm. And once it gets out, it will activate caspases that will kill the cell, right? So the other members that oppose backs and back, that's the BCL2, BCLXX, they don't allow backs and back to form these pores. So they oppose this action of forming those pores in the mitochondrial membrane, thereby making the mitochondrial membrane impermeable to cytochrome C. So the backs, BCL2 and BCLXL, they keep cytochrome C inside the mitochondria, while backs and back try to get cytochrome C out of the mitochondria into the cytoplasm. So that's how they oppose each other, right? So for the extrinsic pathway, we're talking about the intrinsic. This is the extrinsic, so it's coming from outside the cell. All right. So you have the cytotoxic T cell here. It expresses the FAS ligand. All right. That FAS ligand will bind on FAS. Another name for FAS is death receptor. All right. So once FAS binds to FAS, that's or CD95, right? That interaction will activate Caspases that will now lead to cell death by apoptosis. And a disorder of that FAS ligand interaction is called the autoimmune lymphoproliferative syndrome, where there's a disruption of that FAS. So cells don't die. Remember, we said in the if you're attending the immunology, we talked of negative selection in the thymus, where cells that recognize bind too strongly to self antigens are deleted in the thymus. But if that, and this is FAS and FAS ligand interaction is part of that process of deleting those self-reactive lymphocytes. But if this FAS ligand is disrupted, then those self-reacting lymphocytes will persist and they can lead to autoimmune disorders. And one of them is this autoimmune lymphoproliferative syndrome. Okay, so the final pathway is the perforin granzyme B pathway. Okay, and this one involves the release of granzymes containing perforin, and so the cytotoxic T cell and natural killer cell, they release enzyme called perforin and granzyme. So what does perforin do? Perforin will bore holes into the cell membrane of the cell, allowing enzymes called granzymes, all right, you can see them here, to enter the cell, and they will also activate caspases that will kill the cell.